Digestion is a word used to summarize the complex coordination and regulation of a series of processes involved in moving nutrients, water, and electrolytes from the external environment to the internal. These processes include mechanical and chemical breakdown of food into smaller molecules that can be absorbed and used by the body. It also involves secretion, which can mean moving water and electrolytes from the interstitial fluid into the lumen of the GI tract. It may also mean GI tract cells releasing hormones and signaling molecules into the lumen or into the interstitial fluid. And finally, motility discusses the general motion or movement of food through the GI tract from mouth to anus. Of these four processes, in order to optimize digestion, secretion and motility are the most highly regulated, and it is through this regulation that waste is reduced and maximum absorption can be achieved. There are a number of challenges that the GI tract and digestion must face and deal with. Those include auto or self-digestion of GI epithelium, maintaining mass balance, making sure that the amount of water and fluid that enters the body is similar to the amount that leaves, as well as defense from microbes that may enter through the GI tract. In order to coordinate and regulate all of these processes, there are a number of different signaling mechanisms that exist. These include GI peptides secreted into the lumen or the interstitial fluid, hormones that may act over short or long distances, and a subset of the nervous system known as the enteric nervous system, also known as the little brain of the body, which helps to regulate these processes. The enteric nervous system is said to be the little brain of the body, and this is because it can receive stimulation from the central nervous system, as shown in the top part of this image, where sensory information is received by afferent nerves. That signal is transmitted into the central nervous system, and efferent nerves bring the response to their appropriate locations. The GI tract, through the enteric nervous system, can also undergo what are called short reflexes, thereby bypassing the central nervous system, where sensation is picked up within the GI tract, and instead of that signal being relayed to the central nervous system, it is sent to a peripheral ganglion, and the efferent nerve's response is then brought right from that peripheral ganglion to its target, thereby bypassing the central nervous system. Digestion can be divided into three general phases: cephalic, gastric, and intestinal. Gastric phase is the most highly regulated and one of the most interesting to discuss, and so that is what we will be focusing on. But before we do that, we have to mention cephalic digestion, because it actually is involved in setting up or preparing gastric digestion. Cephalic digestion refers to a period of time before food is actually eaten. It is an anticipatory response to a sensation of food or a thought of food, perhaps seeing a meal, smelling a meal being cooked. The thought of an upcoming meal may stimulate the enteric nervous system through cephalic digestion, and this is through those long reflexes that were mentioned before via the vagal nerve. The gastric phase of digestion occurs within the stomach, and the stomach is obviously a very important organ. It has a number of functions related to overall digestion. The first being storage of food, and by storing food, it helps to regulate the motility of food throughout the rest of the GI tract. There's obviously digestion that takes place in the stomach, as well as a defense mechanism that is used to ward off against any microbes that may enter the stomach. As we can see in the diagram here in the middle of the slide, this cutaway of a stomach shows that the inner space, the lumen, is lined by all of these folds and bumps. These are referred to as rugae, and if we look close up in what makes up a rugae, we can see that there are actually tiny gastric openings in these folds. these openings, these gastric pits, lead to these spaces that are known as gastric glands. And gastric glands are a piece of tissue, a type of tissue made up of a number of different types of cells. These cells are listed over here on the right-hand side of the image. This slide serves to highlight some of the important differences and the variety of cells that exist in those gastric glands. You notice that each cell type has a very different form and function associated with it. In a little bit more detail, we see that the cell types in the gastric glands come in one of 6 different types. We see mucus cells, parietal cells, enterochromaffin-like cells, which will be referred to as ELC cells, chief cells, D cells, and G cells. Each cell type has a specific secretion as listed in the 2nd column, and the functions of those secretions are listed in the 3rd column. We can see that mucus cells make mucus and bicarbonate, and these have protective functions to help regulate and protect the GI epithelium from the acidic environment of the stomach. We see parietal cells are actually the acid producers as well as the synthesizers of a molecule known as intrinsic factor. We see the ELC cells actually secrete histamine. We see the chief cells are the cells responsible for producing the enzymes involved in gastric digestion, pepsinogen and gastric lipase. D cells produce a hormone, somatostatin, and G cells secrete gastrin. The functions of each of these secretions, as I mentioned, are listed in the 3rd column. So we can see that the secretions of the mucus cell are for protection, and the stimulation for the release of these secretions is mucosal irritation, as well as just being tonically secreted at regular intervals to make sure that there is always a mucosal layer buffering the epithelium from the luminal contents of the GI tract. We see that the parietal cell's acid actually has a function of activating pepsinogen into pepsin, and the vitamin B12 absorption takes place because of that intrinsic factor that is secreted. Enterochromaffin-like histamine secretions help to stimulate acid secretions. The chief cell's enzymes digest fats and proteins. Somatostatin acts as a negative feedback to stop acid secretion, while gastrin also stimulates acid secretion. And as we can see in this table, the stimuli for some of these other relaying molecules is the fact that we see acetylcholine is a stimulator for the release of many of these, suggesting that the nervous system can target these cells and stimulate their secretion. And we also see that some of the other hormones secreted by other cells help to stimulate some of these other cell types to secrete their contents as well. This process is rather complex, so we're going to take a look at that in a little bit of detail. The initial stimuli can be in one of 2 forms. It may be, as mentioned before, through cephalic phase signaling, through long reflexes, the thought of, the smell of, the sight of food, readying the body for digestion. Or this initial stimulus may also just be food actually entering into the lumen of the stomach. And the presence of amino acids and peptides, as well as the distension, the stretching of the stomach walls, will signal enteric sensory nerves to begin the secretion of specific chemicals in response to this signal. So the vagal nerve input, as well as the food present in the GI tract, will activate these G cells, and as mentioned before, the G cells produce the hormone gastrin. We also see that the vagal nerve input also, at the same time stimulating G cells, also activates the ELC cells as well as the parietal cells to secrete their contents. G cells, as I mentioned, are secretors of gastrin. In response to that initial stimuli, gastrin is a hormone that is released into the bloodstream, and gastrin has two effects. It directly causes parietal cells to produce acid in the lumen of the stomach, and it also indirectly does this by stimulating those ELC cells, shown here, to make histamine. And histamine, in addition to gastrin, stimulate parietal cells to produce protons into the lumen of the stomach, making the stomach a more acidic environment. The way that parietal cells secrete acid into the stomach is an interesting response. Specifically, either enteric nerve stimulation, gastrin, or histamine cause the apical membrane of these parietal cells to incorporate an ATPase pump into that apical side. And specifically, this ATPase pump transports a proton to the lumen of the stomach in exchange for a potassium ion into the cytoplasm of the parietal cell. Please note that the potassium that has been pumped in can then diffuse down its diffusion gradient back out into the lumen of the stomach through open potassium channels. The proton that is pumped out actually comes from the splitting of water. The other product of that splitting of water, the hydroxide ion, is combined with the metabolic waste of carbon dioxide using the enzyme carbonic anhydrase to produce bicarbonate. And bicarbonate is then secreted into that interstitial fluid in exchange for chloride ions into the lumen of the parietal cells. The chloride ion then will diffuse down its electrochemical gradient into the lumen of the stomach, and the end result is a production of hydrogen ions in the stomach from that pump, as well as chloride ions in the stomach, in the lumen of the stomach, because of this second process, thereby creating ions of hydrogen and chloride so that hydrochloric acid can be created in the lumen of the stomach. This acid secretion by the parietal cells has a number of effects. The first is seen here. The acid in the stomach will cause enteric sensory neurons to then stimulate the chief cells to release their digestive enzymes. The first is pepsinogen, an active form of the enzyme pepsin, as well as gastric lipase. Pepsinogen is inactive inside the chief cells because it digests proteins, and if let loose in the chief cells would end up digesting the contents of those chief cells, which would be a negative effect of this enzyme. So therefore, the inactive zymogen, pepsinogen, is secreted, and in that acidic environment, it is activated into pepsin, thereby allowing for digestion of these proteins and, through gastric lipase, the triglycerides that enter the stomach. The acid also has a negative feedback mechanism. It stimulates D cells to create another hormone, somatostatin. And somatostatin has the opposite effect of gastrin and histamine and that vagal nerve input. It, in fact, causes all of these other cell types to halt their secretion of their contents. So G cells, somatostatin causes G cells to stop secretion of gastrin, the ELC cells to stop histamine secretion, the parietal cells cease pumping in protons and acid into the lumen. And in addition to that, we also see that somatostatin causes the chief cells to stop releasing their enzymes. So this negative feedback mechanism helps to coordinate the shutting down of the response to that initial stimuli so that the contents are not wasted in the lumen, that not too much of the secretions go unused, and also this is a way for the cells to consolidate and save energy throughout all of these processes that are listed here. And that is a summary of the regulation of the gastric phase of digestion. We see a sort of positive feedback loop stemming from the initial stimuli causing all of these secretions to make acid and enzymes, as well as a negative feedback loop to halt these secretions once there is enough produced.